Good morning, everyone, and welcome to Texas Hemp Harvesters Association with Prairie View University. We are live, everyone, here at Prairie View University, and we are so excited to announce the start and the launch and opening of their greenhouses and their hemp program. So we have a full 90 minutes today that we will be featuring their research, their labs, and what they're looking to accomplish here within the Prairie View Hemp program. Um, Without further ado, first of all, I want to acknowledge that it is Veterans Day, and we want to thank all of our veterans, those who are in service, those retired, and those that may have been lost in service, that we do honor you on this day, November 11th, and recognizing Veterans Day. And without further ado, I'd like to have a word of welcome from our dean, Dr. Souza, with Prairie View University.
Well, thank you, and welcome everybody to Prairie View A&M University, and specifically the College of Agriculture and Human Sciences on this gorgeous day. And so again, salute to all our veterans, including, I believe, Dr. Parks, appropriately attired and with the U.S. flag in the background. So salute to our vets. Thanks for your service. And I will turn it over to Dr. Rajan, and after his remarks, I'll be glad to come back and provide some remarks on behalf of the college. Dr. Rajan.
Thank you, Dr. D'Souza, appreciate it. And thank you for inviting me to this great occasion. And first of all, good morning, everyone. It is my pleasure to join the occasion today, along with our College of Agriculture and Human Sciences research community, along with the Texas Hemp Harvesters Association. As the land-grant university, Prairie View has a strong focus on agricultural research, as we know. We have a comprehensive and rapidly growing, as well as resilient research community. As you know, despite the COVID-19 global pandemic and disruptions it has caused, we continue to progress very well. And this accomplishment, the start of a new hemp program and the greenhouse facility, is just another demonstration of Prairie View's accomplishments and resiliency. Our Division of Research and Innovation has equally been resilient and have made significant investments in and support in elevating research enterprise, and we are rapidly growing. To give a broader perspective, I'm proud to highlight our significant accomplishments. As a university, the research grant pursuits have doubled, surpassing $200 million the last fiscal year. And the research awards and sponsored expenditures have equally doubled within the fiscal year last year alone. So it is important to note that Prairie View A&M University has a major play in the state of Texas with an economic impact of over $766 million to the state of Texas, creating over 5,300 jobs to the state. So you can imagine the significance of the roles each one of us play for the state of Texas. So it is very critical for the agricultural research community to continue excel in creating new discoveries and innovations that will propel the college and the university as a strong institutions, particularly as a strong research institution in the state of Texas. As the Vice President for Research and Innovation, I look forward to see this unique hemp research focus area to flourish at PV, as this area holds significant potential, not only for scholarly research, but also for commercializable innovations. So I'm excited about that. And our office is committed to provide the support to the college and its faculty and research staff, as well as the students, to elevate the research and innovation enterprise overall. So if you need any support that can make your research better from day-to-day operations to strategic growth, we are here to support. So with that, I'm going to turn it over back to Dean D'Souza.
Wow, that's fantastic, Dr. Raj, and congratulations. You've doubled in one, and my goal, as my colleagues know, I want to double in 5. So it's going to take me 5 years to do what you did in 1 year. So congratulations, and Dr. Rajan has been a strong supporter of the college, so very much appreciate that. So once again, good morning, everybody, and so glad you could join us today. And thanks so much to everybody for organizing this event. And again, those who are veterans, thank you very much for your service to our country. So the other amazing thing is how far we have come as a state in just 1 year since hemp was legalized in Texas. I'll give you just a few statistics that I receive occasionally since I'm on Commissioner Miller's Texas Industrial Hemp Advisory Council. So the last data that I got from them as of September 14th, there's a total of 900 active permits, 5.5 million acres and fields, and approximately 11 million square feet in greenhouses. And that's just in 1 year. 174 unique greenhouses, 1,240 registered facilities, and so on. So a lot of momentum in spite of the fact that much of this took place during the pandemic. So this industry is in its infancy in Texas. And being an infant has some advantages, right? For one thing, we're in a better position to learn from and avoid the growing pains of more mature states. And the recognition that at the end of the day, hemp is a commodity. And like any commodity, it is subject to the business cycle and the product life cycle and the price swings that go with that. So those are things that we have to keep in mind as a growing industry, and how do we avoid some of the mistakes, but by the same token, leverage some of the opportunities that we have to grow and further double and quadruple the amount of research funding and outreach funding in our case, as well as a land-grant university. And at the end of the day, hopefully this translates into some academic initiatives as well in the spirit of the land-grant mission. So coming to Prairie View A&M University in particular, over the years and recently under the leadership of our interim research associate director of agriculture, namely CARC, Dr. Parks, who also is the interim farm director. And that is a strategic move that we made recently to enhance our coordination, mission integration, and further promote our visibility and impact. So over the past year, and indeed going back a few years, we have amassed a tremendous infrastructure and what I will call an intelligence support system led by Dr. Veera Surya. And with this infrastructure in place, we can work together with others collaboratively to facilitate goals such as helping producers generate profits, but also manage risks. From the perspective of the College of Agriculture and Human Sciences, to generate research dollars, patents, publications, partnerships, academic and outreach initiatives, and in collaboration with the Texas Hemp Harvesters Association, with the Texas Department of Agriculture and other agencies and non-governmental organizations to grow a sustainable industry. So one of the things that I want to shed some light on is what are our competitive advantages at Prairie View A&M University, and specifically in the College of Ag and Human Sciences, in terms of helping drive this growth in this new industry of industrial hemp. So to start with our facilities, a 770-acre farm. We have these new state-of-the-art greenhouses, and some of the broadcasting today will take place from these greenhouses. We have well-equipped core labs, so there will be other broadcasts coming out of our core labs in CARC, the Cooperative Ag Research Center. We have a new growth chamber and I'm sure that we'll talk about that as well. A new meat lab, and I'm not quite sure where that will fit in, except, there is this new nutritional technology called precision nutrition. And I believe there will be a role for CBD as part of our medicinal plants initiative and under our food security umbrella to figure out how to utilize food in terms of improving health outcomes and wellness outcomes, and CBD and other medicinal plants could be one such avenue. But together with our facilities, we also have the personnel, the lab support, the farm support. We have industry connections, and we also in the state of Texas have a climate and a long growing season. We also have proximity to shipping channels for international trade. So all of this combines to form a great competitive advantage for us at Prairie View A&M University. The other area I want to highlight is our partnerships. This is one area, the industrial hemp area, where there has been an explosion of interest in partnering with us. They see our competitive advantage in this area. We have a few MOUs in place already dealing with CBD, certified hemp seed, varietal, genetics areas, and more recently in terms of improvement of animal nutrition and health. So I think that is a growing area, and given that we have cattle and goats on the farm, that is an area that we could also look at in terms of partnerships and attracting grant dollars and patents and more revenue for the farm to help us grow in areas that we were not otherwise able to grow up until this point. So partnerships are a new area for us. We're still trying to figure this out. And so what I mean by that is, it's easy to have an MOU, but what comes after the MOU? And so that's where we look for more guidance from the Office of Research at Prairie View and also from the TAMAS system, which we're part of, to help us grow and thrive in this new area. So let me talk a little bit about extension and outreach. So clearly, industrial hemp will be an additional source of revenue for our landowners. And indeed, there's been a lot of demand from limited resource landowners for best management practices, for best seed varieties, and so on, to help them in this new and growing industry. So some of the research that we do will definitely translate to helping them decide on things like the best strains and practices. We also have a new small farm institute that is taking shape on our campus and under the leadership of Dr. Clarence Bunch in a cooperative extension program. And we hope that this will be a platform to provide farmer training, workshops, certification, etc., in crops and livestock, from production to marketing, and including niche crops such as hemp. So it's not limited to hemp, but it could include hemp as well. So this is a platform that we're excited about, and it is yet another way that we can demonstrate impact, relevance, importance to the underserved clients and limited resource landowners. So this institute will be launched with capacity funding, but as with all new initiatives, we look forward to grants and partnerships to obtain the investment to grow and sustain this new institute. Teaching and academics. So around the country, if you've been keeping track, there's new courses in hemp and CBD and other derivatives. There's new curricula, new faculty positions, and this is across the country. We are still not to that point yet. However, we are thinking about a new course about infusing our existing curriculum with niche products such as hemp to provide students with the knowledge that they need to become competitive in the job market, including in niche products such as hemp. Hemp also is consistent with our new strategic plan because it is a product that will enable us to expand our research, academic, and outreach missions in ways that resonate with the landowners and with limited resource clientele around the state. And it is also consistent with our plan of work. So finally, in closing, I want to say thanks to the Texas Hemp Harvesters Association, and especially Hannah Wilner and Andrea Salas for organizing this event. Also from the cause, or the College of Ag and Human Science perspective, led by Dr. Veera Surya, as well as Ashwani behind the controls. And he also leads an interesting and innovative center called the Agro Innovation Technology Center that integrates science and technology. Also, thanks to Stacy Gilly and staff in the Dean's office for logistical support, and our greenhouse staff as well, including the manager, Rafis Brew, and his team. And to Sela and her team in the core labs, thanks everybody. It does take a team to put on a show as good as this. And at the end of the day, I very much appreciate, we have 40 participants, so that's a great attendance. And so I commend you folks for organizing this activity. I hope you have a successful day. I know you will. And I'll turn it back to Andrea. Well, if Andrea is not there, we'll turn it to Dr. Parks. I believe he's next on the agenda. Dr. Parks.
Hey, good morning. Am I able?
Yeah, Andrea is muted, so.
I just unmuted. Am I able to share my screen?
Yes. Go ahead, Dr. Parks, please. Yes. Thank you.
Okay, let me get this. Can you see my screen? Let me say very briefly, the Dean has covered much of what I want to say. So I'm going to echo and say ditto to what he said, but a couple of things. One is thanks to Andre and Hannah for organizing this program. You know, this really is a great program that highlight much of the things that the Dean had just talked about. But I want to just add my little bit to that and the excitement that I have, particularly of the things that's going on in the College of Agriculture and Human Sciences, mostly in the last 8 or 9 months, really. 2020 has been a great year for us in spite of the pandemic. I recall back in the fall of 2018, when Dean Gerard asked me to accept the role as interim executive associate director of CARC. And he asked me at that time, you know, what did I see? And I said, I'm looking for one big bang. I'd like one more big hurrah before I go off into retirement. And well, little did I know at that time, and I had no notion that it would hinge around hemp. You know, I had worked on some regional projects back in the early 2000s where we were looking at hemp as mostly as a biofuels product back in the early, in the Sun Grant initiative that I worked on. But, you know, it's been a fast moving, this has been a fast mover, particularly in the last few months for us. And I think about, you know, looking at some things that we had done roughly a year ago when we participated in Dallas in the first hemp workshop, late October of last year, and where we've come since then. We've come a long ways, and we've got some good momentum going. We're gonna see in a few minutes from some of the exciting things that we're doing here within our research system here at Prairie View, some of what the scientists are doing. I'm excited to look at that. But one of the things that I see is there's so much potential in this. There's so much opportunity in this that we are able to look at. One of the things that Dean Gerard talked about is the partnership. We have signed several of those this year. We've got some more in waiting. A lot of opportunity that I see, and our role in that, I would say this, well, a couple of things I want to say is, as a research institution, our goal is, of course, is to assist with the research. That's what we said. That was in our application last year, early this year, when we made our applications through the Texas Department of Agriculture to get a permit. We wanted to be the research institute. We wanted to provide unbiased information so that those that will use this will be able to make informed decisions. That's our goal, is to be able to do the research so that those who want to become producers, commercialization of the product, then they would have some good research. So one of the first things that we're doing, of course, is the germplasm. And that's what we're going to see probably today. One of our #1 projects is develop the industrial hemp germplasm collection, which goes along, by the way, ties in with what we do with our medicinal plants collection. If you visit our greenhouse, that may or may not show up today in the video, but we have an extensive medicinal plants collection. Hemp is just an addition to that, which is an exciting part of our program today. But I want to just echo what has been said. Excited about that, excited about the opportunities we have, the number of partners that we've added to our portfolio this year, the new facilities, new greenhouse and other facilities that's developing, our core lab facilities. We've added new staff this year, still in the process of hiring others. So I'm excited about the prospects. I'm looking forward to great anticipation with the great things that are going to be happening in the near-term future of what we do as we continue to grow and expand the partnership and to be able to add our bit to what needs to be known and to develop this industrial hemp sector here in Texas and in the world. So thank you all for inviting me to say a word on this program. Thank you for the organizers of this program, and thank you for all of the veterans out there for your service to the country.
Thank you so much, Dr. Parks and Dr. DeSalza, along with our VP of Research, Dr. Rajan. What I want to make sure that we express to all those who are viewing and who are with us today, that Texas Hemp Harvesters Association, our main goal is to be very intentional in how we build relationships. And so we are so honored to be able to have this relationship inside of research with Prairie View University and HBCU right here in Texas, and so excited that the work that they're doing. And so if I didn't say, I'm Andrea Salas with Texas Hemp Harvesters Association, and many of you also know the founder, Hannah Wilner. She'll be joining us shortly in the lab, but we're excited that we're taking today, again, as we've said before, Veterans Day, to recognize one of our research partners, one of the leaders inside of the state of Texas when it comes to hemp research, and that's Prairie View University. So again, thank you. We welcome you. And this is the first live program that we're doing. And so we thank you for joining us today. So we'll go ahead and get started. We're inside of the greenhouse at Prairie View. So just a moment, I'm going to introduce everyone to Dr. Aruna. Hi, Dr. Aruna, how are you?
Hello, good. Welcome everyone for this live program. And we are going to go through the hemp research we are conducting at Prairie View and University College of Agriculture. And just to give a little brief introduction about hemp, as you know, the hemp and the marijuana, they belongs to the same plant species, Cannabis sativa. And before cannabis was declared as a controlled substance, it was growing naturally.
Dr. Aruna?
Levels in cannabis plant was not that high. The maximum THC level found in the natural populations at that time was around 3%, 1 to 3%. Now, you will be surprised to hear today we have data to show you within last 20 years time, THC level was gone from 4% to 20%. Now, this is the danger of the whole species because THC in lower levels can act as the medicine, or it has health benefits, but when it's come to the higher levels, it is toxic and also very addictive. So when it comes to the hemp, now we are talking about the species Cannabis sativa. So once it was declared as banned or the controlled substance, this species was landed in the hands of illegal growers. So their intention was only to develop high THC producing plants, right? And then after we started legalizing the cannabis species, what we have is a complete mixture of genetics. So at Prairie View, what we are doing is doing the very basic research to understand the genetics and then go through the chemical fingerprinting and botanical fingerprinting, and then to identify all these strains with the proper markers, genetic markers, chemical markers, and the botanical markers. So we have a very strong group of scientists work on various aspects, and you will be hearing to them in the next few minutes. And also we have great private partnership developed with the seed producers and the cultivators and the industry groups. And I'm very thankful to the private partners like Hemp Harvesters Association, Hemp Lions, and there are various other seed producers who donated us seeds which we can use for the research. What you see in the background here is the germplasm we received from you all, and we are going to analyze all these plants to get the real scientific data before we identify the best strains for the Texas, and also to the best strains for the end usage. Either it can be used in fibers, it can be used for CBD extract, or it can be used as seed grain or other products. So we'll move to the scientist, Hannah.
Hello, I'm Dr. Peter Ampim, and I'll be talking to you about the seeds that we have received and some of the things that we have observed about the seeds. So if you were to take a hemp plant, hemp has an indeterminate growth pattern. So from this plant here, you can see the lower seeds tend to be mature earlier on compared to those at the top. And so because of that, these plants are typically, the seeds are typically harvested when the seeds are about 70 to 80% ripe. And from this plant, you can see that most of those ones are the ones on the lower side of the plant. So what happens is the top plants will not be as mature as the ones at the bottom. And so when you get the seeds and you just soak them in water, this is an example of seeds soaked in water. The ones at the bottom are your most mature seeds. And then you have a second tier, if you show me the second tier that normally floats at the top. And I'm gonna show you a picture of that right soon on the. Okay, so this picture here shows you how those things that seeds separate out. So if you were to take this as your grade A and grade B, your grade A will be the ones that are in the bottom, and your grade B will be the ones that are at the top. So we came out with this idea that, okay, if the grade B is not so good, maybe we can do something else with it. And so we came out with the idea of growing microgreens, as you can see in that photo over there. So those microgreens are very nutritious. They are actually considered a superfruit because they have a lot of nutritional properties, including flavonoids. And a good example of those would be flavins that are good for they are antioxidants, and they also have anti-inflammatory properties. They also have phytoestrogen and even vitamin E. So what we are trying to do is grow these microgreens and analyze what their nutritional content is and see all the phytochemicals in there that contribute to all the medicinal properties that these microgreens can provide. And if we are to look at this, these are the same seeds here. This will be the seeds at the bottom. Those will be your grade A seeds, and this will be your grade B seeds. And as you can tell, the ones at the bottom with the good seeds, they look a little bit darker in color compared to your grade B seeds. Those will be the ones that are floating. So this is one line of research that we are doing, in addition with our goals to go into the field with the best strains that we determine sometime next year.
Awesome. Thank you so much. We will move along to Dr. Kabroom.
Okay, I'm Dr. Kabroom. My research is on shoot branching. I have been doing research on shoot branching for over a decade in other crops. Shoot branching is very important because it affects the yield of crops. For example, these are the two hemp plants. One developed a lot of branch, the other one didn't branch. So for example, if we want to grow plants for seed, for more seed, then we want these kinds of plant or with at least like 2-3 more branches. But this one, if you can see, the plant is spending its energy on branches, on leaves, so it may not produce enough grains or good quality grains. In fact, fruit growers usually remove the lower branches so that they can harvest good quality, high yield fruits or good quality. So in hemp, there was no research on shoot branching, but there is a lot of variation in the number of branches developed. For example, there are a lot of different types of species here growing, some of them with more branches and some of them with less branches. So research on branching is going to be very important in hemp because this crop is a new crop. No research has been done for the past maybe 40, 50 years because it is recently introduced or being legalized. So we have to do a lot of research to improve the branching pattern of hemp and develop varieties for different purposes. For example, if the economically important part of the plant is on the leaf, then we want to develop varieties that produce a lot of branches, a lot of leaves. But if it is for grains or, for example, for fiber, then these kinds of plants are going to be good. So this is what we can do with hemp. Thank you.
Thank you so much, Dr. Kabroom. All right, you guys, we are moving right along in the greenhouse. And we will now go to hemp plants. Just a second here. All right.
Hello, everybody. My name is Nathan Murphy. I'm with Hemp Plants, and we are working with Dr. Runa.
Give me just a second.
We're good to go?
Yes.
So we're working with Dr. Aruna to help track and research that they are doing. Compliance is a mobile app system that has a app for every user in the chain of custody to track and trace hemp throughout the chain of custody from the farmer to the cell. So we provide the ability for Dr. Runa and his other researchers to use their cell phone to track their plant growth throughout the process using the mobile app, where they can check and track along from flowering seeds, what genetics they put in, how they grow, what they do, whether they branch in certain areas, whether they grow tall and strong, whether they flower early, whether they herm out. So for a lot of the research that's going on, there's just so many variables that we help them be able to track. So one of the things that we want people to be aware of is that with all the research that's going on, the more you use the app, the more research you put into the app, the more information you're going to be able to get out of it. So this helps farmers, this helps seed providers, this helps the researchers. So we offer the ability, and just so for our farmers that are aware, we are offering the Hempliance app. If you are in mobile app to use your Android or iPhone, you can download Hempliance today. And 2020 is free for all farmers in the state of Texas. And we offer the app to be able to track your growth cycle, where you're at, all the way up to selling your product. And we have buyers that are looking at this information so that they know what you've grown, you have proof of life, and you're able to sell your crop at the end of the day. As we know that that's typically the most important thing to farmers. When they're putting seed in the ground, they want to make sure that they get a return on their investment. So not only we're helping the researchers, we're helping the farmers directly. We advise you to check out hempliance.app. So this is our current research. If you look at hempliance.app, you can check us out on the website. We have a commercial up that'll explain a little bit more. If you have any questions, feel free to reach out to us. I'm always available for any questions or answers you may be looking for. In the meantime, we're just happy to work with Dr. Aruna to enhance this research to make sure that we know exactly what's going to work well in Texas and how it performs, because we really don't know what's going on in Texas yet. And that's really what the gist of Prairie View and all the research that they're doing is about. So we look forward to continuing this relationship and to continue to help the farmers in any way we can as we move into the next grow season. Thank you for your time. I'm sure we'll see you soon.
This is what I love about live production. If you notice, we are live in the greenhouse. And so I want to thank Dr. Aruna and his team as we have walked through the greenhouse. Dr. Aruna, just kind of show us the area here. I'll go ahead and switch it back over to you. And then once we leave the greenhouse, you will join Hannah Wilner inside of the Prairie View lab. So Dr. Aruna, give us an eyesight of the greenhouse today.
This is actually brand new greenhouse we got for the Prairie View, and we started developing our germination collection here. Now you will see different sizes of the plants, and most of them are started in the same date, but you see they are responding differently, and the growth rates are different. And also look here. And some seeds came with the virus diseases in it. So we need to study what are these viruses. And we also, although we are growing indoor, we see some pests sneaking into the plant. So we are identifying them also. So it's our researchers have a lot of work to figure out these diseases, pests, and manage the crop in a feasible and successful way. So some strains are finishing their life cycle, but we are keeping them for the further studies of the fibers. And some strains are just keep growing like this. Usually hemp is the perennial species, but with these haphazard breeding programs, the plants started behaving differently. So you see some just behave like animals.
That's it.
Yeah, we are now moving to the.
We are now moving to the
labs.
lab. So you guys, thank you so much. Hannah, if you are ready, I'll go ahead and stop sharing my video and you can go live in the lab.
Mute. All right, we have unmuted, and I believe that we are showing the lab.
Yes, yes.
Unmute. There we go. All right, we're set now. Hi, everybody who's joined. Thank you so much. Happy Veterans Day. First off, we appreciate everybody who served, so we have the freedom to use our experience and our knowledge to cultivate, to research, and to grow this beautiful plant in our country. So first off, thank you to the veterans who create the freedom for us to be able to do these wonderful things. Secondly, I'd like to thank Prairie View for having one of the most robust programs in the state. Everything that you guys have done is just a blessing to the rest of the state. We are here with other researchers inside the lab, so welcome. And I also just want to share with everybody who's on, the value of Texas Hemp Harvesters Association is the relationships that we've already seen flourish. Hempliance was introduced to Prairie View at an event we had earlier this year. And we have a number of relationships, right? The second principle of Texas Hemp Harvesters is to ensure that we have responsible supply chains. And so I just want to encourage everybody who's on to join the association, participate. You too can get these relationships that we've provided to a number of others. Also, we are the largest hemp association in Texas. So if you join us, you'll be joining the ranks of many prominent, successful people who are endeavoring in this new world. So without further ado, I am here in Prabha's lab, which is an excellent resource as the first vet for the cannabis plant. So in this lab, we're going to show you about genomics sequencing. And that's really important because this is how we understand the plant from a genetic standpoint. Also in this lab, they are taking identifiers of the physical external plant as well as the internal cell structure of the plant. So we have some really awesome things to show you. I'm going to turn this over to Prabha and we'll show you the genetics as well as how the plant breaks down. So without further ado.
Hello. At this point, you know, hemp has one species that we use for cultivation, but it's a huge mess. So we use these genetic, molecular, and morphological methods to identify the best strains which are suitable for Texas and also which are suitable for different end usage. Let me show you the molecular analysis first. In molecular analysis, we use DNA and also RNA. We can extract DNA from hemp tissues. And from this DNA, we can read the building blocks of DNA. So we call it sequencing. And here you see an example of a DNA sequence of one hemp strain. We call it strain A. And we can get another sequence from strain B. Also, we can do it for large number of strains. After getting those, we can find different markers. Using these markers, we can get the clusters using the state-of-the-art analyses. The software programs we have and servers, we run these analyses, and from that we can find the clusters of these strains, and from that we can find out the best strain suitable for different purposes and to cultivate in Texas. And also we can use different genomic resources we have. One example is we can use GenBank data. Here's an example of GenBank dataset. We have a good genomic resource from GenBank. From this, we can develop markers for different characters or identification of hemp. This is about DNA. Also, we use RNA. We can also extract RNA. From that, we can study gene expression at different growth stages in different strains. Ultimately, we produce mutants using gene editing tool with our famous CRISPR. So there are a lot of resources for genomic work in this lab. Apart from genomic work, we use morphological analyses. Morphology means external, outside appearance of the plant. For example, this is strain A and this is strain B. From outside, also we can see a lot of differences. For example, the sizes of leaves and the number of lobes in these leaves and the length of the petioles. Like that, we can get large number of characters. We compile those characters and we can also analyze them using different software. Another cool example is the hairs distributed in hemp. In one strain, you see here pointed hairs. In a strain, another strain, let's say strain B, we can see glandular hairs. The density of these hairs are very high. From this, we can compile a huge data set and use this for morphological characterization, that is using outside characters. Apart from outside characters, we can use internal structure or internal cellular characters. For example, here we can use stem cuttings like this and then get the different small teeny sections from this and observe it under the microscope like this. This is unstained, and here we don't see the colors. To identify is it better, we use staining approaches. After staining, we can recognize different layers in the stem. Here outside is the epidermal layer, which we call bark. We can easily peel it off. And the immediately next layer is the bast fibers. And here you see the layer which is stained in blue. We call it hard fibers. We can use it for some different purposes like hempcrete and multiple applications. So we also extract these fibers using chemical methods and enzymatic approach and microbial wrecking method. These are examples of extracted fiber. These are bast fibers which we extracted chemically, and these are hard fibers which we have extracted chemically. And in the same way, we use enzymatic method, microbial extraction method, and check which fibers, which strains give fibers having high heat resistant, high mechanical strength. And this is another example of fibers. We treated it using different methods. These are treated with bleach, and these are treated with hot water. And we compare which strains and which methods give high mechanical strength and which strains are high heat resistance. And those are the cool things happening in our lab.
Thank you, Prabha. What a wonderful, wonderful demonstration. We're turning this over to Salah, who has another lab in the same building. She's going to take over from here and show you a really neat research piece of equipment that they have over there. So Salah, go ahead, take it away.
My name is Salah. I'm managing the core lab of the research center. And it has 3 sections, which is molecular biology, microbiology, and analytical chemistry. And here where I am now, it is part of is molecular biology and part of is analytical chemistry. So when we get samples, first we have to know what we want to analyze. For example, we want to analyze heavy metals in the plant, or it can be the microbial content of the plants, or it can be, it's not only chemical, it's like pesticides, okay? Pesticide, insecticides, or it can be what is the content of the plant. So we can do all these works in this lab. So we, because we do have these 3 sections, so every part of the samples, it goes through different sections. So the first thing we do is, we wanna, when we get the sample, we wanna dry the sample and extract or actually digest. It depends on the type of analysis. So for example, this instrument, it does both digestion and extraction. What we do is we measure the plant, and we put specific amount of sample in this machine, and we add the chemical, the solution, whatever solution we want to use based on what we're looking for, and we extract. And the good thing is it gives us fast results. I mean, once we have this one, we use different instruments for if we want to know the content of heavy metals. We have in the other side of the lab, which is called ICP. It's gonna give us the content of all heavy metals, whatever we're looking for. And if we wanna know the pesticides or insecticides, then we have the machine in here right there. It's called GCMS. It's gonna give us the concentration and the type of the pesticide or insecticide. The good thing is we do have library in the software, so it is exactly tell you what is in it. So after we do that, if we want to analyze the component of the plants, we do again digestion, and we can do any type of analysis. It can be using HPLC. It can give us, again, the concentration and the content of the plants. So we do a lot of different kind of analysis. Yeah, I can show you some of the data we have.
Go ahead.
Oh, okay. So can you see? This is an example of the graph which we analyzed it recently, and when we are clicking here is giving you the library. The library is telling you what is in it. You see, that's what exactly, it gives you the formula and the chemical name. And if there are other kind of synonym names, it gives you all those choices. So it's very, really, I mean, you cannot miss anything from here. So we can analyze the component of the plants for safety reason, the microbial content. We can do that, even including sequencing. You know, like a hemp, it can be contaminated with mycotoxin or endotoxin. We can analyze all those toxins too. So it is really, we can do everything, whatever we need for the analysis.
Thank you, Salah. We really appreciate your information and showing us all these great labs. For everybody who's on the call, if you haven't gone to college, I highly recommend Prairie View. Their facilities are top notch. They have everything that you could want, and they've got the most brilliant professors, doctors, etc., who are teaching courses and teaching everybody how to be the most excellent version of whatever they're doing. So with that, I believe that we may have some questions in the chat. We were waiting for questions until later. Until afterwards. So if you have questions, we encourage you to put them in the chat. Yes, we did. We did not. Where's Dr. Sutton?
She was on. She has a
Ah, okay. So then, I'm sorry, apologies. We have one more presentation from a lab.
From Dr. Sultana.
Dr. Sultana is on, and we will switch to her, and then she will show us that. Thank you, Sultana.
Good afternoon. Thank you very much. In fact, my presentation will be a PowerPoint because I am at a different location. So I'm sharing my screen. Okay. So thank you very much. It's a great opportunity for me to share my research with others in this great event. In fact, I am thinking about how can we apply all these hemp in the medical field or in biomedical field. My background is chemical engineering and food process engineering and biomedical engineering. So my focus will be how we can use these hemp fibers or hemp extract for the medicinal uses. So if I want to share that a slide about a burn injury, if we consider this case, like if someone has skin injury, we know that if it's 2nd degree or 3rd degree, we need a temporary support for the healing process. So normally for the healing process, we use, or the doctors use autograft, allograft, or xenograft. But there are always problems with morbidity or limited availability, immune response or rejection or pathogen transfer. So nowadays, the scientists are making the artificial skin grafts or membrane that can actually help help in wound healing process. So here, actually, this picture shows how the surgeon use an autograft or an allograft. So they peel the skin and then they actually use on the wound site. But actually, no one likes this type of reoperation because it's painful. So we are trying to make synthetic tissues or synthetic skin graft so that we can avoid these autograft or allograft. So here is my actually research focus, that how can we prepare membranes in the lab, and then we can culture the cells to examine the toxicity, and we can also actually check if it can really help in the real life. So here one of my project where we actually used polymers to use the membrane, and we actually characterized and actually we characterized those membranes. We can see these are all nanofibers. So these are called scanning electron micrograph. So these are like nano or microscale fibers, and we like these fibers because these fibers has the similarity of our native tissues that can actually help the healing process. So we did other characterization. I will not speak too much technical things, but here we can see that there are actually, we have very good microscopic analysis technique to actually characterize the membrane or the substrates. So we can actually know that if the membrane or the fibers are hydrophobic or hydrophilic, because we need hydrophilic fibers for this for the cells to grow. We actually culture the cells to know if it is the toxic or not, because it's a very important criteria to be used as a biomedical like implant or anything. We have to know the if it is toxic to cells. So we do the live dead cell assay using human fibroblast cells, and we found that around the viability was 95%. All these green colors shows the actually that the cells are living, not dead. If it would, if those are toxic, we could see all the cells are in red color. So also we can actually evaluate the antibacterial properties. So here we used E. coli and the Staphylococcus aureus to examine if the membrane can actually inhibit the bacteria. So here you can see that we actually use the zone inhibition technique. So we can actually use these, we can say if this membrane can have antibacterial properties or not. And also we actually, in another project, we used silver nitrate nanoparticles. We actually coated the fibers and checked the antibacterial properties of the membrane. And also we can actually, in another project, I fabricated the cancellous bone-like structure. So you can see that the cells, actually these are the cells. You can see that on the surface of membrane or scaffold, we can see the cells are growing. That means these membranes are favorable for the cells to grow. So here I am working at undergraduate medical academy as a research scientist, you can see our students are actually fabricating the membranes using some technical methods. So it's called a freeze drying or lyophilization technique. Here you can see that these scaffolds are porous structure. We also use, we can make some thin films using polymers. So here we used chitosan and pectin to check the antibacterial properties to be used as skin graft. So here one of our students who is doing very good in her research, and I received 2 grants from VPR to help the students to continue their research. And also I found that these hemp materials can be used in several ways. So it can be used as fillers, or it can be used to reinforce the polymer composites. And also the most important thing, that the hemp possesses antibacterial properties that actually can inhibit a wide range of pathogenic bacteria. And also we can actually understand that or more experiment will be needed, that these properties are coming from perhaps from the cannabinoids or the alkaloids or the phenolic compounds of lignin. So I did some research, and I found that several groups in US, Australia, and in other countries, they are actually trying to use this hemp as biomedical application. So some main areas are wound dressings. As I told you that in my research, we can use these hemp fiber for the wound dressing application, skin tissue regeneration, and also as a drug to treat bacterial infection directly. And also it can be used to purify waters. So sometimes the water can have some metallic or some other things that we want to remove. So we can use this membrane as a filter. And also we can use this hemp extract in dentistry or food packaging because it has antibacterial properties. It can be used as food packaging and also as food storage or prolong the shelf life. So here I am just mentioning some of my scholarly contribution. I am an editor of 1 book and author of 2 other books. So I have published around 100 journal papers and also book chapters and several conference proceedings, and those were cited more than 1,200 times. And also here are some of my selected journal papers. But not only the scholarly activities, we can actually commercialize all these hemp based products that can be used for the biomedical application, also for the food processing and food preservation technologies. So that's all for today. Thank you very much.
Thank you so much, Dr. Sultana. We are back at Prairie View at the Cooperative Agriculture Research Center with Dr. Parks, Dr. Aruna. I'm Andrea, and this is Hannah. We really want to thank everyone for joining us. Again, this is our first time, right, guys?
It's the first time.
This is our first time.
Program.
Exactly, but it certainly will not be the last. And we're so excited to what Prairie View has to offer. Hannah, you said it so very well about if you're considering a school, whether you're continuing your PhD or you have a student that's coming, that's looking for a college, Prairie View is certainly the place. So Hannah, do you have anything you'd like to share with our guests?
I think we've already given an excellent overview of the program that is being conducted, has started here. They have so many plans for future expansion. So if there are any questions, please email us. We will get them to Prairie View. Again, this video and the YouTube link will be in the members' resources. So all members will have access to this at any time. And ultimately, we just want to thank you for tuning in today during Veterans Day. We appreciate you spending your lunch hour with us and learning. all about the new research and what's happening with genomics and identifying the best plans for Texas. So Texas industry can take off. Dr. Parks, Dr. Aruna, any last comments?
Just let me say thank you, Hannah and Andrea, for putting this together. This is a great program. You know, couldn't have done it without you. So we appreciate so much for you putting this together. Hey, thank you all for tuning in today. Now the secret's out. Now you know what we have. This is a snippet of what we do at Prairie View. The best is yet to come, as we say at Prairie View. Prairie View produces productive people. The work that we're doing, we're just in the beginning, early stages of it. So great things we anticipate coming out of what we do. So thank you all today. Look forward to having you on other programs in the future.
Awesome.
Yeah, I need to thank my researchers team, and they did great job. And we are waiting to hear from you. If you have any questions, you can go to our websites and shoot an email. We are open for any discussions to answer your questions. And also, big thank goes to our IT team.
Thank you, IT.
Mr.
Thank you,
Ashwani. Made it possible. And this is the first time we had this kind of program. And we are planning to improve it a little more and more smooth. Thanks everyone for joining. And
so
I think you are muted.
So is Dr. Jawar coming back on?
Is he still on?
I think he's on.
Oh,
Dr.
yeah.
Jawar, there you are.
And our wonderful dean to give our final sign off.
Wow, what can I say? I'm so awestruck by all of the great things going on at the university, and particularly in the College of Ag. But you know, it's great to see all of the unexplored uses of what I suppose could be a miracle crop in many ways. You know, from feed to food additives to the medicinal uses, and now onto material science. But there was a really good question asked by our colleague Joyce Osborne about any current research using hemp to inhibit COVID-19. So Dr. Aruna, can we get some research going in that area? Because then we could be the new Pfizer and so on, and you know, live beyond our wildest dreams, as it were. So that would be a great area to pursue. I don't know if there's anyone who's doing any work in that area at Prairie View or anywhere else, but that is something that could be the future of hemp. So this material science aspect of it, I think, is really lucrative for us. So thanks everybody, and thanks to Dr. Rajan for joining us, and to all the rest of the crowd who were able to share part of their day with us. And the Texas Hemp Harvesters Association wish you much success in your endeavors to make this more of a lucrative crop for Texas landowners and growers. And thanks again, everybody. Stay safe, stay well, and salute again to all the veterans out there. Take care.
Everyone, thank you.